Now we are going to study about specialized or restricted transduction and how is it different from the generalized transduction. So there are certain temperate phages transfer only a few restricted genes of the bacterial chromosome to the recipient bacterial cell. The transfer of bacterial genes adjacent to prophage only to the recipient chromosome is called restricted or specialized transduction. This was discovered by Morse in 1956. It is made possible by an error in the lysogenic life cycle of the phage. When a phage genome is introduced into here you can see here, when the phage genome is introduced into the bacterial chromosome as prophage, here you see this phage DNA integrates into the chromosome of the bacterial chromosome, and here it can exist as a dormant for several years. So this is known as prophage. When this prophage deintegrates and picks up a piece of bacterial chromosome, it uses the enzymes which excises the bacterial and the viral genome, and here this part of the bacterial genome is transmitted or integrated with the viral genome. Replication of the viral DNA and destruction of the bacterial DNA. Now the virus uses its machinery along with that of the bacterial machinery and releases its extra chromosomal DNA, where here you can see the red part is the viral genome and blue part is the part of the bacterial genome, or restricted part of the bacterial genome. So here we have this transformed or integrated bacterial genome along with that of the viral genome. Now the virus capsid synthesis and assembly. The next step is the synthesis of the viral protein along with the capsid, which encloses this integrated new recombinant viral genome, which consists of a few fragments of the bacterial genome as well. So upon induction, the DNA becomes free, containing a small segment about 5 to 10% of bacterial chromosome. It multiplies, disintegrates the bacterial chromosome. After assembly of phage DNA plus bacterial chromosome, the phage is released from the bacterial host. Here you can see this, that after their lysis, first the formation of the capsid, the viral protein, along with this integrated new genome containing the bacterial genome as well. Now this new type of phage is released and it infects the another bacteria. Usually this phage is defective and lacks some part of its attachment site. When this defective phage insects a bacterium, it introduces DNA containing a piece of the bacterial chromosome. The defective phage cannot reproduce without assistance. The gene of the phage can insert with homologous DNA of the infected bacterium. Now here this phage again uses the bacterial machinery for homologous recombination, and then it incorporates its DNA into the new bacterial genome. And you can here we find three types of genes: the bacterial, the viral genome, and the new type of the bacterial genome as well. So sometimes crossing over occurs between the homologous gene loci of the bacterial chromosome and the donor DNA attached with the phage genome, resulting in the integration of the donor DNA with the recipient DNA. Now, how is it different from the generalized? It does not lyse quickly. It is capable of surviving of several generations. Now, due to detachment from bacterial chromosome, a small portion of bacterial DNA remain attached to the viral DNA and packaged into the new capsid. Now, the most important point, these are closely related strains which show similar sites, whereas in generalized, less specific bacterial strains are used, but its frequency is quite high and no progeny is produced. Whereas the frequency of the specialized transduction is low, but prophages are formed.